Hello students, myself Jinalisha welcomes you on behalf of Saraswat International Academy. Today we will begin with the next part of the chapter, Principles of Inheritance and Variations. Students, in the last lectures or in the previous videos, we have studied regarding Mendel's monohybrid experiment. So basically the experiments which deals with the inheritance of one genes, they were called as the monohybrid experiment. Right. Now from the monohybrid experiment, Mendel derived certain conclusions, and this conclusion today we know it in the form of the law of inheritance. So basically from Mendel's monohybrid experiment, he derived two important conclusions. One conclusion was the law of dominance, to which he gave basically there were two exceptions. One was incomplete dominance and another one was codominance. So these two also we have studied in our previous videos. And another important conclusion derived from the Mendel's monohybrid experiment was what? Law of segregation, or also called as the law of purity of gametes. So now students, today what we will be dealing with? We will be dealing with the inheritance of the two genes. So students, those experiments which deals with the inheritance of the two genes, what are they called as? They are called as dihybrid experiment or dihybrid crosses. What are they called as? Dihybrid experiments or dihybrid crosses. Which means here we will be studying the inheritance of two characters simultaneously. In this particular experiments, we will be studying the inheritance of two characters at the same time. So students, what is a dihybrid cross? So a breeding experiment which deals with the two characters at the same time. What is it called as? A dihybrid cross. So basically a cross which considers the inheritance of the two characters at the same time. We will study the inheritance of the two characters of the two genes at the same time. What is it called as? It is called as a dihybrid cross. Now which plants Mendel selected for his dihybrid cross? So basically, of course, he had selected pea plants. Now which two characters together he studied in pea plant? One character was the color of the seed and another character which he studied was the shape of the seeds. Which two characters he studied? Two characters. One was the color of the seeds and another one was the shape of the seeds. Now students, Mendel already performed monohybrid experiments for these two characters. What Mendel already performed? He performed monohybrid experiment for these two characters. From his monohybrid experiment on these two characters, he found that students, yellow color, it was dominant over the green color. It means yellow color is a dominant character and green color is which character? A recessive character. Similarly, when he performed the monohybrid experiment on shape of the seed, he found students that round shaped seeds, they were dominant over the wrinkle shaped seeds. So this character is which character? A dominant character. And this character, the wrinkle shaped seeds, is which character? The recessive character. So by performing monohybrid cross, he concluded that when he studied color as a character, he found that yellow color dominant over green. And when he studied seed shape, he found that round shaped seeds were dominant over the wrinkle shaped seeds. So now students, what Mendel did? He took a plant with both these dominant characters. Mendel took a plant with both these dominant characters. So, he first of all concluded what? The round and yellow color are dominant characters and green and wrinkled are which type of characters? Recessive character. So, now he performed a cross. What type of cross? How did he perform the cross? So, he took a plants which were having yellow color and round shaped seeds. So one of the parent selected by Mendel were dominant for both the characters. Which both characters are we talking about? One is color of the seed and another one is the shape of the seed. So one plant he took that a plant which was dominant for both the characters. That means he took a plant with yellow color and round shaped seeds and he crossed that plants with have the green color and wrinkle shaped seeds, which means recessive for that character. He took a plant which was having a yellow color and round shaped seeds and crossed this plant with a plant having a green color and wrinkle shaped seeds. Now students, when he performed this cross, he found that all the plants in the F1 generation were having what? Round and yellow seeds. He found that all the plants of the F1 generation were having round and yellow seeds. Now when Mendel selfed the F1 plants, when he selfed the F1 plants, on selfing the F1 plants, we get the plants of which generation? F2 generation. But students, here he obtained 4 different kinds of plants in the F2 generation. When he crossed or when he selfed, basically when he selfed the F1 plants, he obtained 4 different kinds of plants in which generation? The F2 generation. Now students, we will cross the plants. So basically, plants with the pure characters, pure dominant character was crossed with the plants with the pure recessive characters. So basically this RR stands for what? It stands for which structure? Now see students, the R stands for the R genes. They stand for the shape of the seeds. And Y genes, they stand for the color of the seeds. So basically he crossed the round, basically this is round, this stands for yellow, this stands for green, and this stands, this stands for wrinkled and this stands for green. Now students, what are the gametes produced? Here all the gametes will have R and capital Y, and here all the gametes will have small r and small y. Now students, what would be obtained in F1 generation? So all the plants would be capital R, capital Y, small y. Now here we know that capital R is dominant over small r. This conclusion came from the monohybrid experiment. So capital R stands for what? Round. Capital Y is dominant over the small y. What does capital Y stand for? Yellow. So all the plants produced in the F1 generation were having what type of structure? Capital R, or which type of genes? Capital R, capital Y, small y. Now students, he selfed the F1 plants. So basically this same structure again. Now students, where will we write the gamete in the Punnett square? They are written on the left corner columns and on the top rows. Where are the gametes written in the Punnett square? They are written on the side of the left columns and on the top rows. Now students, what different combinations for the gametes we obtain? See students, which will be the gametes form. The gametes for this and this will be same. So first of all, we will write the gametes. So it will be capital R with capital Y, capital R with small y, then small r with capital Y and small r with small y. So from this particular plant, how many different types of gametes are formed? 4 different. Capital R, capital Y, capital R, small y, small r, capital Y, small r, small y. Similarly, same gametes will be produced here. Capital R, capital Y, capital R, small y, small r, capital Y, small r, small y. Clear, students? Now cross them. So students, how will we cross them? First of all, we will cross this with this. So what will be obtained? Capital R, capital R, capital Y, capital Y. Clear? Same way, now we will cross this with this. So again what we obtain? Capital R, capital R, capital Y and small y. Now crossing this with this, what will we obtain? Capital R, small r, capital Y, capital Y. Lastly, this with this. So what we obtain? Capital R, small r, capital Y, small y. Now student, in this way we will find the genotypes of this. So on crossing this with this, see students, what we need to take? This and this. So we obtain capital R, capital R, capital Y, small y. This with this. So what we obtain? Capital R and small y. Now this with this, we obtain capital R, small r, capital Y, small y. And lastly, this with this, we obtain capital R, small y, small y. Clear, students? Now same way, this with this, we get capital R, small r, capital Y, capital Y. This with this, here this two, capital R, capital Y, small y. This with this, small r, capital Y, capital Y. This with this, small r, capital Y, small y. Finally, the last one, this with this, capital R, small r, capital Y, small y. This with this, capital R, small y, small y. This with this, small r, capital Y, small y. And finally, this with this, small r, small y. Now, these are the 16 different combinations obtained in the F2 generation. Now, if we study the phenotype of each and every plant, now we know that capital R is dominant over small r, that is round is dominant over wrinkle, and capital Y, that is yellow color, is dominant over what? Green color, that is small y. So here, if you study the phenotype, here capital R, students, what will be? It would be round. Capital Y stands for what? It stands for yellow, right students? Same way we will write the genotype. Here also it is which type of genotype? Round and yellow. Here also capital R is dominant over small r. So it will be round and it will be yellow for Y. Same way here both are different but capital R dominant. So here also we obtain round and yellow. Now student, here also tell me, capital R and capital R. So of course it will be round. Capital Y, small y. Which is dominant? Yellow. So yellow colored seeds. Here capital R, round. But students, which y is there? Small y. And both are small y. That means which colored seeds it will be? It will be green colored seeds. Similarly, here we get round and yellow. Here what will you obtain? Capital R, small r. It means round. Small y, it means which color? Green color. Same way, here what you will obtain? Capital R, small r. So it means round. Capital Y, which means yellow. Here also round, and this will stand for yellow. Here small r. So when small r is there, which condition will be developed? Wrinkled. So students, this is wrinkled, and capital Y stands for yellow. Similarly, again this is wrinkled, and here it is yellow. Same way, here round, yellow. This is round. This too stands for green. Here wrinkled. This stands for yellow. And finally this is wrinkled, and this stands for what, students? Green color. Now students, see here, we will study the phenotype ratio. What is the phenotypic ratio obtained here? So students, now you will see different combinations. You will see the round yellow combination, then you will see the round green combination, then you will see wrinkled yellow combination with the wrinkled green combination. Now you calculate these phenotypes. Round yellow. See, this is round yellow 1, this is round yellow 2, 3, 4, 5, then this is 6, then 7, 8 and 9. So basically, how many plants? 9. Now round and green. So this is 1, this is 2 and this is 3. Now wrinkled yellow 1, 2 and 3. And wrinkled and green only 1. So this is the phenotypic ratio of the dihybrid experiment. What is this? This is the phenotypic ratio obtained from the dihybrid experiment. Clear, students? How many different? See, I told you, when he selfed the F1 plants, he obtained 4 different kinds of the plant in the F2 generation. Which were the 4 different plants? The phenotypes were with the plants with the round and yellow seeds, with the round and green seeds, with the plants with the wrinkled and yellow seeds and plants with the wrinkled and green seeds. So these were obtained in what ratio? 9:3:3:1. Whereas when we studied the monohybrid experiment, what was the phenotypic ratio obtained, students? It was 3:1. But when he performed the dihybrid experiment, what phenotypic ratio he obtained? 9:3:3:1. So students, this is regarding the Mendel's dihybrid experiment. Now from this Mendel dihybrid experiment, Mendel he got the conclusion. Now students, I told you from Mendel's dihybrid experiment, he gave a very important conclusion, and that conclusion is in the form of law of independent assortment. So basically students, you have to remember that from Mendel's monohybrid experiment, two laws were given, law of dominance and law of segregation, and from Mendel's dihybrid experiment, law of independent assortment was formed. So students, these are the three laws of inheritance. First law of inheritance, which is law of dominance. Second law of inheritance, that is law of segregation, and third law of inheritance, that is the law of independent assortment. Now students, what this law states? It states that when two pairs of traits are combined in a hybrid, that is when there are two characters in the hybrid, segregation of one pair of character is independent of the segregation of the other pair of characters. So students, it states that when we study the inheritance of two characters simultaneously, then segregation of one pair of character is independent from the segregation of the other pair of character. But students, keep in mind that this law states true only for those chromosomes which are located on the different chromosomes. Keep in mind, this law of independent assortment is true only for those characters whose genes are located on the different chromosomes. Say for example, I consider human beings. How many chromosomes are there in human beings? 23 pairs. So is it like that only 23 chromosomes are there? So only 23 genes will be there? No. There are lot many characters as compared to the genes. So students, what about the inheritance of those characters whose genes are located on the same chromosome? Listen my question very carefully, students. I told you the law of independent assortment. What does the law of independent assortment states? It states that when two pairs of traits are combined in a hybrid. So when in a hybrid there are two pairs of characters, then segregation of one pair of character is independent of the other pair of character. Right? Now I told you this law states true only for those characters whose genes are located on the different chromosomes. But we know that number of characters are far more as compared to the number of chromosomes. So students, what about the inheritance of those characters whose genes are located on the same chromosomes? So students, for this law there is an exception, and that exception is called as linkage, and it is through linkage we can study the inheritance of the genes located on the same chromosomes. So what is an exception to the law of independent assortment? The exception to the law of independent assortment is linkage. Now students, we will study the law of independent assortment. So students, when we perform the dihybrid experiments, we got two new phenotypes, right? What were the original or parental phenotypes? a plant with a round and yellow seed, and another was a plant with the wrinkled and green seeds, right? So these two, round and yellow, and another one is the wrinkled and green. These two were which phenotypes? Parental phenotypes. But when he performed the dihybrid experiment, he obtained two new phenotypes. Which were the two new phenotypes? Round and green, and another one was the wrinkled and yellow. So these were the two new phenotypes obtained in the F2 generation along with the parental phenotypes. Which were the parental phenotypes? Round and yellow, wrinkled and green. So along with the parental phenotype, he found two new phenotypes in the F2 generation. Which were those? Round and green and wrinkled and yellow. Now the ratio he obtained was 9:3:3:1. What ratio did he obtain? 9:3:3:1. Now how he got this ratio and how the gametes are formed? Now students, to study this we have formed that the Punnett square, it can be effectively used to understand the independent segregation of two pairs of genes during meiosis and production of the eggs and pollens in F1 capital R, capital Y, small y plant. Now students, which type of the gametes will be formed and how the genes behave during the meiosis. So if we want to study the independent segregation of two pairs of genes during meiosis and what type of eggs and what types of pollens are produced, if we want to study the segregation of the two pairs of gene during meiosis and what type of eggs and pollens are produced, this can be easily studied using the Punnett square. Now students, say for example in this particular F1 plant, right? This is an F1 plant, right? Now in this F1 plant there are how many pairs of genes? Two pair. One pair is capital R, small r, and another pair is capital Y, small y. So first of all we will deal with the segregation of one pair of gene. Say for example we consider one pair as capital R and small r, right? In this particular plant, suppose we consider one pair as capital R, small r. So we will consider the segregation of one pair of genes, capital R and small r. So now students, according to law of segregation, we know that 50% of the gametes will have the capital R gene, 50% of the gametes will have small r gene, right? Suppose if we consider this pair of gene, which pair? Capital R and small r. So here 50% of the gametes will have capital R gene and 50% of the gamete will have which gene? Small r gene. Now students, each gamete having either capital R or small r, which allele they will also have? They will also have the allele capital Y, small y. See here, which is the another allele? Suppose this is the allele. So here 50% of the gametes will have capital Y and 50% of the gametes will have small y. So now students, a gamete beside having capital R and small r, it will also have which allele? Capital Y and small y. But students, you should remember one very important thing, that segregation of this 50% of capital R and 50% of small r, it is independent of segregation of 50% capital Y and 50% small y. It means that segregation of the genes related to the shape of the seed is independent of the segregation of the genes which stands for the color of the seeds. They do not have any relation. Segregation of seed color and segregation of the seed shape. They are independent of each other. That is, these two genes will segregate on its own. These two genes will segregate on its own. Both of their segregation are independent of each other. The segregation of the color of the seed is not dependent on the shape of the seeds, nor the segregation of the shape of the seeds is dependent on the color of the seeds. So students, what is my conclusion from here? That the gametes having capital R and small r genes, they will also have what? Either of the capital Y or small y. Why? Because segregation of 50% of capital R and 50% of small r, it is independent from the segregation of 50% of capital Y and 50% of small y. So we can say that 50% of the capital R, 50% of the small r having gametes will have capital Y gene along with it, and 50% will have the small y. So here what we can say, 50% will have the combination capital R and small y, and 50% will have the combination small r and small y. Similarly, here also we can say that 50% of the capital R will have capital Y with it, and 50% will have capital R with small y. So students, how many different genotypes we obtained? 4 different genotypes of the gametes. Which are those? Small r capital Y, small r small y, capital R capital Y, capital R small y. So here you can say that 4 different types of pollens and 4 different types of the eggs are formed with the genotypes capital R, capital Y, small y, then small r capital Y and small r small y. Which are the 4 types of the genotypes? Capital R, capital Y, capital R small y, small r capital Y, small r small I, with each of the frequency 25 or 1/4 of the total gamete. So now by forming the Punnett square, what we can know? We can easily derive the composition or genotypes of the zygotes of the F2 plants. And to this there is an exception which is called as the linkage. So students, this is an important conclusion derived from the dihybrid experiment, and that is the law of independent assortment. Students, law of independent assortment holds true only for those genes or only for those characters whose genes are located on the different chromosomes. But we all know that there are many genes located on a single chromosome. So what about the inheritance of those genes? And that can be explained through the linkage, and thus linkage is an exception to the law of the independent assortment. Now students, we will study finally, after conclusion of the monohybrid and dihybrid experiments, we will study through the table the results of monohybrid, dihybrid and trihybrid cross. Monohybrid cross, the cross which studies the inheritance of a single gene. Dihybrid cross, a cross which studies the inheritance of 2 genes at the same time. And trihybrid cross is a cross which studies the inheritance of 3 genes at the same time. So students, here you can see the table. Here number of traits or hybrid, that is number of characters studied. Then is the experiment, that is whether it is a monohybrid, dihybrid or trihybrid experiment. Types of the gametes formed. That is, how many gametes will be formed. Types of the zygote formed. Number of the phenotype, number of the genotype, the phenotypic ratio and the genotypic ratio. Now suppose number of characters studied is 1. So basically it is which type of cross? A monohybrid cross, right? So say for example the genes are capital A small a cross capital A small a, or it may be written as capital A small a. Now students, the type of the gametes produced will be 2 raised to n. So here how many characters are we studied? 1 character. So how many gametes will be formed? Only 2 gametes. Recall your monohybrid experiment, you will see that only 1 gamete will be formed. Then the number of the zygotes, it will be 2 raised to 2, that is basically 4. So 4 different combination you obtained in the monohybrid experiment in the F2 generation. Phenotype, number of the phenotype, only 2 combinations were there. So 2 raised to n. Here how many character? 1. So total 2. Number of genotype formula is 3 raised to n. That is 3. Recall your monohybrid experiment. In the genotypic ratio what you were obtaining? Capital T 1 time. 2 times capital T small t and 1 time small t. So total how many genotypes we obtain? 3. Phenotypic ratio is 3:1 and genotypic ratio is 1:2:1. Now students, a dihybrid cross, that is number of the characters studied at the time are 2. Types of the gamete formed 2 square 4. Remember your dihybrid experiment. Number of zygotes or offsprings 14. When we obtained the Punnett square, 14 offsprings were there. How many phenotypes were there? 4. Round yellow, right? Then round green, wrinkled yellow, wrinkled green. 4 phenotypes. Genotypes total 3 square, 9. Phenotypic ratio is the square of this, that is 9:3:1. Genotypic ratio is again square of this, 2:4:2:2:1:1:1:2:1. And finally, students, again this is for knowledge only, the experiment studying the inheritance of the 3 genes at the time, that is trihybrid cross. So gametes formed will be 2:3, 8. Offspring produced will be 8 square, that is 64. Phenotype will be 8. Genotype will be 27. Phenotypic ratio will be cube of this and genotypic ratio will be again cube of this. So this is the results obtained from the various crosses, monohybrid, dihybrid and tri. So students, today we have studied regarding the Mendel's monohybrid experiments, and a very important conclusion derived from it was the law of independent assortment. We have also studied that. We have also studied the results obtained from the various monohybrid, dihybrid and trihybrid crosses. So students, today I end my lecture here. In the next lecture, we will be dealing with the next part of the chapter. So students, thank you very much. I end my lecture here.